Hello students, so welcome back to the channel. In this video, we are going to study about incomplete dominance. Beta, again try to recall the Mendel monohybrid cross, okay? And just try to recall the law of dominance. According to the Mendel, in dissimilar condition, okay, in dissimilar condition, only one allele will express. And the one which is expressing, that was called as dominant. And the one which is not able to express in heterozygous condition was called as recessive. So according to the Mendel, in dissimilar condition, of allele, only one allele is able to express. And the one which is able to express, that was called as dominant. Clear, beta? That was called as dominant, and which is not able to express, that is known as recessive. But in post-Mendel era, according to this, what we observed, according to incomplete dominance concept, in F1 generation, the allele was not able to express completely. Means F1 hybrid does not resemble either of the parent, does not resemble either of the parent. Is it clear? I'll just give you an example, try to understand. See here. If you remember, according to the Mendel concept, when the tall plant was crossed with the dwarf plant, F1 generation was only tall. Remember, beta, this was F1 generation, which is only tall, and this was parent. But in case of the incomplete dominance, what we are finding, when the red flower is crossed with the white flower, the F1 generation was not resembling any parent, and you were getting only pink color. This is F1 generation, and this is pure parent. So according to the Mendel, one gene was completely dominant, another was completely recessive. Okay, so Mendel was able to discover only one type of gene interaction, that is dominant and recessive. But after that, when we applied this law of inheritance on the another plants, we found that independent dominance is not always working. Sometimes the F1 hybrid is intermediate, and we find that it is incomplete dominance. Now see the book. So see, beta, when experiment was done on the pea plant, they were doing it by using different traits, other traits also. It is found that sometimes the F1 had a phenotype that does not resemble either of the two parents. Means F1 hybrid is not resembling with any of the parent phenotype. F1 had a phenotype which does not resemble either of the two parent and was in between the two. And such type of inheritance is known as incomplete dominance. Clear, beta? And the example in which they have studied, that was good example is a dog flower, also known as snapdragon or Antirrhinum species, is very good example to study incomplete dominance. So you can see through the diagram, when the red flower is crossed with the white, okay, the gametes always, you know very well, homozygous will produce only one types of gametes. So all the F1 generation is appearing to be pink color. They're not resembling any of the parent, and F1 hybrid is intermediate between the two. Is it clear, beta? And when F1 is going for selfing, this is a result when the F1 is going for selfing. So it is given here also, when the F1 was self-pollinated, then what we are finding, that the ratio is changing. According to the Mendel, F2 generation ratio was 3:1, but here phenotype ratio is changing to 1:2:1. Is it clear? Let me show you on the one note. So now see beta, when this F1 generation was going to selfing according to the Mendel, what we were finding in the F2 generation, we were finding 3 tall and 1 dwarf. Clear beta? But here when the F1 generation is going for selfing, we are finding the phenotype ratio of F2 generation as 1 is red, 2 is pink, and 1 white. The phenotype ratio, this is the phenotype ratio of F2 generation is modified. Phenotype ratio of F2 generation is modified. Now for genotype, let us use the symbol. Here I'm using for capital T, this symbol, and here for dwarf. So F1 generation is this one only. Now when you are doing the selfing, you have to make a Punnett square. So let us make the Punnett square here easily. It will be easy for you. So you will make it gametes here. And this gametes here is male gamete and female gamete. And finally, you are putting it this with a genotype. Clear beta? So your genotype ratio is coming as 1 homozygous tall, 2 heterozygous tall, and 1 homozygous dwarf. So this is the genotype ratio of F2 generation. Same let us do with the incomplete dominance, that flower color. This is a symbol for red and this is symbol for white we are using. The pink is heterozygous. Now when selfing, so obviously we have to make the Punnett square. Clear beta? So we are making the gametes here. Male gamete and then female gamete this side. This is red. This is pink. This is pink and this is white. So what you will write for the genotype ratio here, you will write 1 homozygous R, 2 heterozygous R, and 1 homozygous white. Okay, this is the genotype ratio of F2 generation. So if you see carefully from this, please focus better. According to the Mendel dominant recessive relationship, we are finding phenotype ratio is 3:1. Clear beta? But according to incomplete dominance, the phenotype ratio is 1:2:1. So your phenotype ratio is modified from 3:1 to 1:2:1. Is it clear? According to the Mendel, what is the phenotype ratio? Once again, beta, it will be 3 tall and 1 dwarf. Means 3:1. But according to incomplete dominance, what is a phenotype ratio? We will say 1:2:1. Now, what is a genotype ratio? Genotype ratio is not changing. Genotype ratio, according to Mendel, also it was 1:2:1. And according to incomplete dominance, also it is 1:2:1. Is it clear? So, in the short summary, we can say Mendel observed only one type of interaction in which F1 resemble only one parent. Which parent? That is dominant parent. But in case of incomplete dominance, F1 does not resemble either of the parent. And the phenotype is an intermediate. Is it clear, beta? Now let me give you one question on this so that your confidence can come more. Now let us see, beta. Suppose I'm saying taking this one, dihybrid, okay? This is a dihybrid and I'm saying it is going for selfing. Now, question is given to us that this is showing dominant and recessive relationship. So I'm writing D for dominant, R for recessive. And they are giving us this one. This is showing incomplete dominance. It is showing incomplete dominance. So I'm just writing it this way. I hope you are understanding it. And they are asking us what is a phenotype ratio when it goes for selfing. So now see beta how to solve. You know very well when this is dominant recessive, it means like a Mendel. Okay, beta. And phenotype ratio according to the Mendel was, F2 generation was 3:1. Are you getting me, beta? You will get the phenotype ratio what? 3:1. But you know very well, right now we studied incomplete dominance, and according to incomplete dominance, what is a phenotype ratio you should get? 1:2:1. Isn't it? This much is clear now, beta? According to the dominant recessive, your phenotype ratio of F2 generation should be 3:1. And according to incomplete dominance, your phenotype ratio should be 1:2:1. Now, how to find for both? You have to just multiply it. Now, do one thing, multiply 1 with this 3 and 1. So your answer will be 3:1, beta. Then you multiply this 2 with both. Then you will write 6:2. Then next you multiply this one. Then you will write 3:1. So your phenotype ratio will be 3:1:6:2:3:1. Is it clear? When they are asking you dihybrid cross in which one allele pair is showing dominant, another allele pair is showing incomplete dominance. So this is one question which I tried to give you based on both types of interactions. I hope this topic is easy for you. I tried to make it easy for you. Thanks for watching.